includes every kind, class, genus, species of the plant Cannabis sativa. These are materials to be used in macroscopic and microscopic process. First is our cutter, push pin, the ruler, our puller, our yarn dropper, and also the tray and our compound microscope. Also our saline solution, our diluted HCl, our chopping board, and also the tape. And these are the substitutes that we use for our Cannabis sativa. First is the lady's finger leaf, the camphor leaf, the papaya leaf, the bitter gourd leaf, and our eggplant leaf. And now, we will identify the ventral part of our papaya leaf. Identify our papaya leaf. The ventral part of our papaya leaf. The tip, the margin, the vein, the midrib, our leaf base, our petiole, and also our axil. And now, we'll proceed to the dorsal part of our papaya leaf. And as we see, the parts of it is the same in our ventral part. And now, the venation type of this leaf is pinnate, and the margin type of this leaf is entire. Compound of this leaf is palmately compound. And now, let's proceed to the second specimen, which is the eggplant leaf. The first part of it is the tip, the leaf base, our margin, the midrib, the vein, our venules, and the leaf base, the petiole, and also the axil. And now, let's proceed to the dorsal part of it. And as we see, from the dorsal part, it's just the same in our ventral part. And as we see, the venation type of this plant is palmate, and the margin type of this plant is entire, and the type of this leaf is simple. Now, let's proceed to our next specimen, which is the bitter gourd leaf. Part of it, or the ventral part of it, is the tip, the margin, the vein, the midrib, the vein, the leaf base, the petiole, and also the axil. Now, we'll proceed to the dorsal part of it. And as we see, the ventral part and the dorsal part is just the same. And the venation type of this plant is palmate, and the margin type of this plant is serrate. The type of leaves of it is a compound, which is a palmately compound. And for our next specimen is our lady's finger leaf. And now the ventral part of it is the tip, the margin, the midrib, the leaf base, our vein, our petiole, and also our axil. Dichotomous, serrate, and palmately compound. Now we have the camphor leaf. Its venation is pinnate, and margin is serrate. The type of leaf is simple. Now we will proceed to the elimination part of our specimens. The first one to be eliminated in the specimens is the eggplant leaf. The second one is our camphor leaf. And the specimens that will continue through our microscopic process is the lady's finger leaf, the bitter gourd leaf, and our papaya leaf. And now we'll proceed to our microscopic process, which we will examine the leaf with or without chemicals. Now we'll proceed to our microscopic process, which we will examine our first specimen. It is the lady's fingers leaf and without chemical. And as we see to our compound microscope, it has a transparent trichomes. Now, let's proceed to our first chemical, which is the diluted HCl. And now, as we see into our compound microscope, our leaf was burned because of our chemicals, and we have a lot of veins that we can see. And now, the same process, but we will use the saline solution. And as we see from our compound microscope, we can clearly see the transparent trichomes and the veins of it. And at this moment, we will see the discrepancies that with or without chemicals, and this one is without chemical. And the next one is with diluted HCl. And the last one is with saline solution. And now, let's proceed to our next specimen, which is the papaya leaf. And now we can clearly see the veins, but we can see its trichomes. And now we will proceed to our first chemical, which is the diluted HCl. And with our diluted HCl, we can clearly see its veins, but we can see its trichomes. And now we'll proceed to our next chemical, which is the saline solution. And just like our first chemical, we can clearly see the veins, but not its trichomes. At this moment, we can clearly see the discrepancies between the three. Without chemical, with diluted HCl, and lastly with our saline solution. And for our last specimen is our lady's finger leaf. And without the chemical, we can clearly see all of the trichomes. And now let's proceed to our first chemical of it, the diluted HCl. And for our first chemical, you can clearly see some bubbles, the veins, and it doesn't have a trichomes. And to our last chemical, which is the saline solution. And as we see from our compound microscope, we can clearly see the veins, but because of the chemical, it was burned. And also, we didn't see some trichomes. At this moment, you can see the discrepancies between the three. First one is without chemical. The second one is with diluted HCl. And the third one is with saline solution. And now, let's proceed to our unknown sample. And now, the first one is without chemical. And as we see from our compound microscope, we can see the cystolytic hair trichomes. And now let's proceed to diluted HCl. And now we can clearly see the veins, but because of the chemical, the leaf was burned and we didn't see some trichomes. And for our last chemical is the saline solution. And for our last chemical, we can see some trichomes, but we can't identify what kind of trichomes it is. Moment, we can clearly see the difference between the three. This without chemical and our with diluted HCl. And lastly, with saline solution.
And now, as we reach the end of our discussion, I would like to briefly summarize what we have learned about the substitutes and adulterants of cannabis sativa. Among the leaves we examined, one plant stood out for its closer resemblance, the lady's finger plant, or Abelmoschus esculentus, commonly known for producing okra, as leaves that can sometimes appear similar in shape to cannabis leaves due to their lobed structure. While they are not identical, the lady's finger leaf shows a closer visual similarity compared to many other leaves, especially when viewed quickly or without detailed botanical analysis. In conclusion, understanding the substitutes and adulterants of cannabis sativa is important for accurate plant identification in scientific study. Through careful observation and comparison, we learned that although many plants may look similar, the lady's finger leaf is among the most visually alike to cannabis leaves compared with other leaves we observe. This reminds us of the importance of studying plant morphology closely before making conclusions. Thank you for listening, and we hope this presentation has helped you better understand the similarities, differences, and the importance of accurate identification when studying plants like cannabis sativa.